Hi everyone. Every protein in our body, no matter how complex, begins with a simple building block, that is the amino acid. Let's take a closer look at the tiny but powerful structure that makes life possible. So when we talk about amino acid, we first will discuss the backbone of the amino acid. So the amino acid has an alpha carbon. This is an alpha carbon. And then we have, to the right side, we have a carboxylic group. And to the left side, we have an amino group. Upwards, we have an H. And then we have an R group. Now, you all must be wondering, what exactly is this R group? So, this R group is actually a side chain. This could be anything. And this is responsible for determining which amino acid this particular amino acid will be. So it is very essential for the classification of amino acids. Now when we talk about amino acid, as I said that this is the main backbone of amino acid, right? So, in every amino acid, this is going to be the same except for this R group. So R group is going to change, but then this backbone is going to remain same. I'll be showing you how is it possible. Now, let us say if I have a protein, right? So whenever you see a protein, if I say I have a protein, right? So this is like the folded form of protein. If I make it in a linear form, then whenever you see a protein, you'll see that at one end it has an N terminal, and then to the other side it has a C terminal, right? C terminal. So you must now understand that this particular N is actually the NH2, and this particular C is actually the carboxylic group or functional group which is attached to the alpha carbon, right? So this is the reason why whenever we draw a protein, one side of the protein is termed as N-terminal or N-terminus, and the other one is known as the C-terminal. Now let us see some amino acids so that you could be more clear with what I'm trying to say. So let's take an example of one of the most simplest, that is glycine. So glycine, let us take blue, So as I said, the backbone is going to be same for all the amino acid. So this is the backbone, right? And then in place of R, at first I used to write R, right? But then I'll replace it with something which is known as H. So instead of R, I've replaced it with H. This particular amino acid is known as glycine. This is the most simplest and the most flexible amino acid found. Now let us take an example of another amino acid which is known as alanine. So the same thing we're going to repeat. We're going to draw the backbone of amino acid, which consists of the carboxylic group, the amino group. This is 2. The H group. Now we have, instead of H, we have an CH3, a methyl group, right? This one is known as alanine. Another one, let's take another example. Same carbon. We have the carboxylic group, the amino group, then we have the H group, and then we have the C. We have CH3, then we have CH3, right? As you can see that this carbon is not able to fulfill its valency because it has 3 bonds. So we're going to add one H over here. Now it has fulfilled its valency. So now as you can see that this is the R group and this is the backbone, right? And this particular amino acid is known as valine. Now, the thing is that in valine, if you see the structure, the name itself is very much associated with the structure because the name valine has an R group which has an inverted V, right? So if you see here properly, you'll see that this particular R group has an inverted V, right? So this is the R group. So there are many amino acids, many more amino acids. So if I'll be very much particular about, oh my god, if I'll be very much particular about the amino acids, then amino acids in our human body, there are around 20 standard amino acids. Now you must be wondering, only 20, or like, do you think there are more? Yes, there are many more than hundreds of amino acids, but then the only amino acid that is like coded or that is translated into the proteins of humans are these 20 amino acids, right? So you must be familiar with the process of transcription and translation. So these 20 amino acids gets translated during the process of translation, and other amino acids, they do not usually gets incorporated into the proteins that are finally made after this translation. So I hope you understood this video. If you did, please give a thumbs up, and then please like and share this channel. Thank you.